Hello and welcome everyone in another episode of Researcher Celebrity powered by Empowering Science Foundation. Today we have Dr. Dhananjay Chaturvedi with us. Dr. Dhananjay Chaturvedi, you will just experience during this conversation that what he brings to research, but a brief introduction about him. He has done his bachelor's in microbiology from Garwari College, Pune, and then master's. He went to developmental biology and pursued at TIFR Mumbai. For PhD, he moved to USA and did his PhD in UT Southwestern at Texas. Then want to serve back to society. And in his words, happiness lies in family. So he moved back to India, joined as postdoc in NCBS Bangalore. Presently, he's a senior scientist at CCMB Hyderabad, which is a CSIR institution. With this brief introduction, we would like to welcome Dr. Dhananjay.
Thank you very much. Thank you.
Yeah, so let's start the conversation with our trademark question, opening one, that how and when you decided that you want to be a researcher?
I actually decided to get into science in 9th standard. So I would do, and I think much of that has to do with my circumstances. My father also is a scientist, he's a theoretical physicist. I used to see that he's happy just doing what he does. You know, he's if given a chance, he's happy with a pencil and paper and writing equations and solving things. So that was quite the inspiration. The other thing is I actually liked animals a lot, as a lot of young children do. I think an important inspiration for me was my teacher in 6th and 7th standard, science teacher, who would teach from the textbook, but force students to think just by asking very simple questions. So taken together, I think the decision was made then. And that made taking up both mathematics and biology in 11th and 12th standard as a student in Hyderabad very easy, because at the time, in Hyderabad, at least in the opinion of aunties and many uncles, you grew up either to be an engineer, a doctor, or a failure. I did not want to be the first two. So I think I've done okay since, though, So I think to answer your question, it was that decision was made for me fairly early on.
So when you talked about the motivation and inspiration coming from dad and teachers, was that the time you really wanted to be a researcher, or because you were clear that you don't want to be one of the two, neither the doctor nor the engineer, and the third option which you gave was the failure ones. So it was a very strong brain. It needs a very strong brain for a kid to say that, okay, I'll try something else, even if they call that as failure.
Yeah, so I mean, that's what I'm saying. To me, looking at the happiness in my father and his colleagues, I did not count as being a doctor, non-doctor, or non-engineer as a failure at all. It was just the perception
Nice.
of people around me. And so I actually didn't think that I was going to, I didn't want to do something different. I actually wanted to get into science. And I was genuinely interested because things, in 9th standard, you learn things like mechanics, how you can tell everything about a moving object along its trajectory. I think that's fascinating. It's very powerful. And solving problems has always been something I like to do. So getting into research, getting into science is something I really wanted to do. Having said that, my first exposure to research was somewhat traumatic because that was, I think I can say that I got into actual research properly during my master's. And proper research is very different from Discovery Channel. It is very different from, the books that are popular science books that are written. So it was, I think I grew into it. And my perception of research and what it takes to do research was formed during my master's. So what didn't really change. I didn't want to do anything different. I really wanted to do science. And so I got this KVPY fellowship early on. that gave me the confidence that I'm okay at this. Maybe I can, proceed. So yeah, so that the aim was clear early on.
Yeah, great. So this is for the audience that even presently, the Empowering Science Foundation believe in breaking the barrier. Dr. Dhananjay Chaturvedi, people like him are the reason who believe in breaking the barrier, and they do break the barriers, because even now in India, if you are taking biology, the first expectation of society as well as parents is that, okay, we want you to be a medical doctor. If you are taking the mathematics and physics chemistry, then they want you to be an engineer. Although it is changing, but still this is to go thing. And when a kid at ninth standard want to do something else, because for him happiness is the driving force, and for many obvious reasons, that is the thing. We got a great researcher back in India. So talking of research, when you moved for, you said that you tasted the research, the first traumatic experience, as you said, in masters, when you just got introduced to it, still you chose to pursue a PhD. Why was that?
I mean, once your motivation is set, you want to see it through. You want to take it to the very end. If it doesn't work out, it doesn't work out. And a PhD is something that you absolutely need for any sort of research career. So even though it was somewhat traumatic, I think there are different levels of trauma. This was not that traumatic. So it was a learning experience. I still wanted to do research, right? And honestly, at the time when I was reading about stem cells and their potential and applications, it was just fascinating. And I think I was very lucky to get into TIFR, and that gave me exposure to all kinds of very interesting research that was being done in DBS at the time, from some very powerful, investigators who are active even today in DBS and TIFR. So I think, although I did not, although relative to my peers at the time, I didn't find as much new. It didn't deter me from continuing elsewhere, because if it doesn't work here, maybe it'll work elsewhere, and you pursue that hope. So you apply far and wide for PhD programs based on what your interests are, and see where you fit in, and then take that next step independent of what happened previously. Just take what you learned with you to the next step. So that's how I basically decided to continue with a PhD, though MSc was not easy.
Nice. And this is something which we also try to tell our audience and viewers here, that the idea of research is not to marry with the concept or the project. You are a researcher, you have more things to do. And if some place or a particular topic is not working out for you, doesn't mean that you still have to stick onto it or to give up research as all, because you should align your interests, and then whatever the fields we believe that you are good at, and then you really want to get trained in that also, because what we have seen is some researchers are great at philosophy. Most of them are, actually. That's the reason why they called as Doctor of Philosophy. So they enjoy reading history, but they are great at biology. So you have to demarcate all of this. So Dr. Dhananjay, when you moved to United States, and before already you made one change from microbiology to developmental biology. Not many are courageous enough to do that. Then from microbiology, what was the topic you did in your PhD, and how was that right?
Yeah, so in my PhD, I was actually looking at chromatin modifications affecting germline stem cell function. And we were doing this in Drosophila ovaries. So the basic idea is that you have a stem cell that is capable of self-renewing and differentiating. What are the factors that control it? Drosophila are a beautiful model system in which you can answer these questions, because the kinds of manipulations you can do in Drosophila are not as readily available in vertebrates, for example. And also at the time, to tell you the truth, somebody told me that you get papers quickly in Drosophila. They didn't say that, you know, the projects have to work as well. So that's what I did in my PhD. I had done rotations in neurobiology labs chiefly, in fact. So I'd done a rotation in Luis Parada's lab, in Jenny Shea's lab, and then I came to this lab because Mike was just such a powerful speaker, and when he described the problems that he was addressing, I really liked the problem, I mean, I got offers to join all the labs. In fact, there was one other lab that offered me a position as well. But I think the problem was at the time was very, very interesting. And that's how I got drawn to Drosophila. And honestly, very quickly, if you have some sense of how problems are answered in biology, you'll realize that Drosophila to vertebrate biology is like mathematics to physics, you know. It's much cleaner, you have much more control, and you can answer some very, very difficult questions with little or no ethical problems, you know. So I think all of those things, the problem, the model system, and in my case, the PI, drew me to what I decided to do for my PhD.
So then when after this thing, because you said the inspiration was your father, who's a developmental physicist, the theoretical physicist, and you moving to biology. So what were how these conversation goes at home?
It was, you know, it wasn't about science at all, because the conversation at home was, you won't mostly. So my father did not. So my father's word of advice on this was, just make sure that you choose a line to work in where somewhere in the middle you don't ask yourself, now where have I landed? You know, you should like to do it enough such that even in difficult times you can stick with it. And my mother had a very much more practical approach, because she said that, look, people who have done engineering are earning well, they are you know, they're comfortable, and you can't just be happy with, you know, science and results. If somebody has to pay bills, there'll be aspirations from people around you. So so that was her advice. And also, I mean, of note, there's a difference between theoretical physics and experimental science.
Yes.
You know, could be chemistry, could be biology. There's a huge difference in so your capability to imagine systems and work out their rules is what is good for theoretical physics. You can imagine all you want in biology, but until you have experimental proof, it doesn't amount to much. So there was that difference that I discovered, like I said early on, that you know, research in biology, experimental research is very different. But at home, overall, the idea was split. But I stuck with what I thought was important at the time. It was that you are happy doing what you do. And as time went on, I realized that not only do I like teaching, people around me learn when I teach. That's a good thing. And so, so basically, there's no reason to suggesting to me that I should move out.
Yeah. Absolutely. And this is one thing which we always say, that do whatever makes you happy. If research is what makes you happy, Absolutely. There are no reason you should put brakes on. Just keep your leg on accelerator. Just don't crash, because nowadays we do see a lot of crashes in research too. That being said, so now when you are in a lab which deals with chromatin and then iPS, like not iPS, is the stem cells. After that, during the PhD, did you already think it through as you started with that you really want to be in research for rest of your life?
So that actually never changed, especially early on in PhD. The idea was that um, I would work hard. I would, publish papers and go on to do a postdoc in a good lab, publish more papers, and then start research in India. That was always the idea. And but, you know, as you realize in a PhD, that plans are useless. So, if a PhD teaches you anything, it's that you can plan all you want, things will unfold the way they will. Um, and, in my PhD, I actually ran into projects that were somewhat uh, hard to, say, make a story out of to publish. I'll tell you, as far as the audience is concerned, I'll do my best to explain it in simple terms. So we were looking at one protein and we thought it was a homolog of a human protein. So that this protein in Drosophila germline stem cells also had an equivalent functional protein in humans, right? Now, if you take away the protein by one method, which is RNAi, you get a reproducible phenotype, right? The next, how do you know that the effect taking away this protein has on these cells is because of that protein? You put back the protein. So this is called a rescue experiment. And if putting back the protein takes away the change, whatever change there was because of this protein. You can take the human protein and stick it into Drosophila germline stem cells that don't have your protein of interest, and it would still alleviate the change. So the human protein was rescuing things as well. So I thought that this is good, this is very nice. But when we generated now in biology, you have to show the same thing through multiple approaches, just to be sure that it's not the approach that is giving you the effect. So when the other way to remove a gene is to, or a protein is to take away the gene. And when you generate a clean removal of this gene, knockout, it had no effect. It did not look like what was happening using this knockdown method. Now, so we are satisfying all genetic formalisms, and yet it was hard to show or be certain in that time period what to explain what we see. It was hard to do that. Yeah. So, I mean, but then you keep trying and trying, right? So, and by the way, we didn't have tools for any of this available. I had to make the antibody, I had to make the RNA lines, I had to make the knockout in flies, which was considerably laborious relative to what it is today. So it took a while, but when you end up somewhere in fourth year and fifth year with results that are hard to explain, nobody comes to your rescue. Nobody says, I think you did a good job. It just becomes a reflection that you are still unproductive. You have not published, So I would say that around towards the end of my PhD, I genuinely considered, or at least asked, what am I good for other than research? Because up to that point, it was always research. And those let's say, so many circumstances conspired such that at the time, not only did I not have a paper, even the big guys, big labs did not have funding to take people on. So even if I convinced them that I'm a good candidate, they didn't have funding. And so I'm very grateful to Vijay to this day, because for somebody, because he, I had written to him because I was genuinely interested in the problems that were being looked at in his lab. He was looking at a muscle stem cell population. The paper had come out just the year before I had graduated. It was a beautiful study, and I wanted to see what implications that has in adults. And when I wrote to him, he did invite me to do a postdoc with him. And so here, I again count myself lucky 2 times that I get a third chance to sort of continue and use what I have learned in the process of research. So yes, so there was that lag in enthusiasm to pursue research when things got real, because you need a job. You need, you're close to 30 years old, and you're still, sort of getting a degree. So but again, I will say this, all through towards the end, if your intention is to do research, you have to give it the best shot and see where it takes.
Absolutely. And this is one thing which just Dr. Dhananjay made it even clear every single time we have this thing, that research is not about just the rosy beds. Mostly it is about the thorns which you have to walk on, sometime to sleep on. So if you are committed to it, give all the thought before committing that you want to be a researcher, you want to pursue a PhD, because nothing has changed in decades for a PhD student. It was tough 50 years ago, it is tough today. The only thing which has changed is people coming up with more and more innovations to solve their problem. As Dr. Dhananjay just mentioned, that few tools were not there. Now the tools are available. You can do the study of 10 years in 2 years or 3 years max at max, the available tools. So the tools will be there, but in research, the research question is the most important part. What are you asking and what are the implications? So Dhananjay, when you came back from United States, joined Professor Raghavan's lab. Now, as you said that this was the third chance, you were lucky enough. But the truth is you were passionate always. Nothing matters that how traumatic or how how many troubles are there in your way. But you were like, I am not going to give up. So how was the experience at NCBS?
I think it was fantastic for me. To start off, I started trying to look at the role of insulin-like peptides in the stem cell population that gives rise to muscles and how that affects their growth. And then parallelly, like I said, I was interested in looking at what these stem cells do in adult muscles. And parallelly, Rajesh in Vijay's lab was already addressing that question. So the interesting bit was the original project that I was doing was was interesting. It would just take a very long time to get results that would mean anything. So in short, the juice was not worth the squeeze, right? So parallelly, I had been trying to look at how can you identify this adult muscle stem cell population in fruit flies. And I got, again, well, let's say, there was some amount of, so there was a lot of thought put into ways in which we can find such a marker for the stem cells. And then again, I tried a large number, of which one clearly worked. And and then we were able to show that it is specifically these cells and their progeny that respond to cell injury in muscles. Luckily, I mean, soon after we published, there was a group in Cambridge that published very similar results. And so, independent of our studies. So we were very, it was gratifying to see that we found something for the first time, because the existence of such a population was never reported in flies. It was thought that they don't exist. So we had conclusive data to show something new. That was quite a thrill. And after that, Vijay is one of those amazing mentors who gives you all the freedom and all the responsibility. You do what you want to, but it's your responsibility, right? To sort of survive in this environment. You know what you have to do. Go do it. So once this came through, we actually were continuing to trying to understand how injuries affect muscles, right? And can you induce an injury in a muscle and see and look at repair? Now, the trouble is the tools available for looking at repair rely on looking at one plane of the muscle. The muscle is a three-dimensional object. How can you be certain that what is true for one plane is true for the rest of the muscle? So lucky again, in Sanjay, sort of was spearheaded the thing of getting a micro CT scanner on campus, on NCBX campus, and we were allowed to use it. And though we didn't have the set protocols to use the micro CT scanning to look at soft tissue in flies, they sort of dried up the tissue. If you're looking for injury repair in muscles are these, wet, large tissues. If you dry them out with alcohol, they shrink. Any changes or repairs won't be noticeable. So we were able to design a way to look at micro CT scans or Drosophila muscles using micro CT scans and get complete, accurate, three-dimensional images of 800 micron objects to 0.5 micron resolution. And that has been immensely useful for me since then. In fact, we went on to try this on different insects like bees. Turns out people from campus, then started using this on every sample they could get. And I think many of these studies are valuable, like getting insect species from the northeast of India, where there's remarkable diversity in habitat and conditions. Then what happens to the internal structures? You cut the animal, you destroy the tissue. So what does it look like inside? And so our method is there as well. So, and NCBS has this, gives you this freedom where you, as a postdoc, like for example, Raja, we had this undergraduate teaching, a system where between, I think in the spring, right? Because before the summer vacations and exams, we'd sort of screen a group of undergraduates and we would teach lecture series. And that would be based on the interests of postdocs on campus, their expertise. We would knit it together in a way that encouraged students to think. That was our primary focus. And the wealth of smart people are in the Bangalore area, at least amongst the students who applied, is just immense. And so it was a joy teaching in this lecture series. And then the Postdoc Fellows Association gave me this space to solve one problem that I think still has not gone away in India. And I'm sure any postdoc in India will appreciate this, which is that if you have a postdoc from India, there's always a doubt if you are good enough to hire in academic positions within India. So this is something that a lot of my colleagues have felt, I have felt during the course of applications. Because very few places go straight to to your research proposal.
Yes.
Right? You filtered out because for other reasons. So we needed a forum where postdocs within India could come together, share research with themselves and potential employers. Earlier, it was just academic employers. And then we decided, well, the world is a big place. We have more to contribute. And so the postdoc symposium caters to more than just academic jobs now. And we've basically, the progress of a postdoc's career is at the heart of their activities now. I have nothing to do with it anymore. So, but then the point is, had there not been support from the higher ups within NCBS at the time, this would not have come about. I would not have met, colleagues or future colleagues. We would not have had a sense of the reality on the ground, because what you don't find out working hard in your lab towards a paper is that there is much more,
Yes.
because so you can have impact factors and such, but they will invite you to the interview. After that, your impact factors don't matter.
Yes.
It's what you propose to do, what your research program is going to do. We had no idea of these things because at the time. So overall, I think my postdoctoral experience in NCBS was great. There are many factors: a very supportive mentor, a very supportive environment, smart people all around me. I think that made it good for me.
Nice. So we'll come to the mentor and mentee question now. But here I would like to mention, as because you brought it up now. So for all the postdocs in India or abroad who want to join either academics or industry in India, postdoctoral symposium is a must attend event. You should always look forward to it, because this is something which is not offered anywhere, all I can say everywhere. And NCBS provides you with this idea, which is a brilliant initiative. If you want to go out of your lab, because normally people go for meetings, workshops, conferences, that is different. But this experience is a wholesome experience where you can have way more than interactions and the way it goes. So now coming back to Dr. Dhananjay. So you mentioned about all the great mentoring you got. And you were mentoring during the postdoc time also. Now, in the full role of a mentor, how these researchers have evolved from as a mentee to a mentor?
Well, so I mean, again, one thing I didn't mention is that during my postdoc, I got to work with a fantastic group of people who wanted to do their dissertation work or just wanted to train in the lab. So I think the 7 odd MSc theses that I was able to bring to fruition in Vijay's lab gave me a sense of the student group I will work with, what their needs are, how much attention, how do you speak to people, and things like that. So that was very important. So luckily, Sharashij and Megha, who are in my lab right now, I think it's been a productive interaction. So talking to students, I haven't forgotten what I was as a student, what I needed. And so I apply the same, that thought process informs how I interact with people. So transitioning from a postdoc mentee to a PI mentor, in that sense, has not been that difficult, touch wood. I hope it stays so. But what has changed is now I'm responsible for a lot more than my own career, my own little corner of research. I not only have to sort of ensure that the day-to-day running of the lab is smooth, that everything that people in the lab want to do their research is made available and smoothly. The lab environment has to be good. Going forward, the people in the lab need to pick up skills, hard skills that will be useful not only within a research environment, but in industry. The soft skills that are essential in every sort of environment, and how to be versatile, things like that, and basically help them become independent researchers. They do whatever they choose. So that transition it's been a little scary, right? The science part, even in my entire life, I think that very often science is not as difficult as dealing with the people who do science. And I'm socially awkward a little, so it's been a learning experience. And overall, I'm still learning. I'm technically a baby as a PI. It's been 6 months, so yeah, infant. Okay, I'll have my annaprashan soon. No, just joking. So But yeah, it's a learning experience and I'm looking forward to it. I'm happy with the way things are at the moment and I hope to continue. This intermediate fellowship that I received recently ensures that we are good on funding for the next 5 years. So that should not be an obstacle. I think sort of driving oneself to answer questions cleanly, at every step, being honest with yourself and the data, and to answer questions. That is something that we can do now, without having to worry about pennies. So yeah.
So congratulations for the and I forgot to introduce within that, because DST sub intermediate fellowship is something which I believe that is not very easy to get. It is a tough fight. And now coming back to your communication and scientists, okay, you said people like science, because hard skills and soft skills, this is something which is hard skill you can teach a student about the basics of science, the concepts, but giving or inculcating those soft skills, that how they present their data, how they interact to a person who does not belong to science, so who has no idea what housefly is, or how Drosophila is different from housefly. For them, they are just flies. And when you have to convey your research to them, the key of soft skills is very important. And mostly and broadly, scientists are known as bad communicators. When you say that you are socially awkward, and when you present your data nationally, internationally, and when you are having these conversations with the students now, how do you see that the communication is going presently in the research field?
So depending on where you are, there may be a thrust on communication, oral, written, for different audiences, or they may not be. Depending on where you are, you may have people around you from whom you can learn these skills, or you may not because they're not there. My feeling is that, in fact, it seems to be my general experience, that a lot of people have some very, very interesting findings, but they're not able to convince anyone else that they are interested. And
Yes.
to find out, and so how do you develop a method for communicating to any audience, knowing that audiences are different? How do you develop that to communicate your findings and research? Because, and so here I think it is critical to know how good you are at communication. Try explaining to people at home who do not do research. Yeah. If they retain what you are saying, you are communicating the big picture.
Yes.
And so you can start with those things at home. And at the next level, when you want to put in a little more detail, like I said, we've got some very smart students within the country who, given time and experience, might be at a level with you. So they are your next audience to whom you can not only talk about the big picture question, but also the method that you approach to get to the problem.
Yes.
Right. And further, the implications of what you do requires experience to assess. And so that is your peer group. And I think incorporating, making a presentation, bearing in mind every bit of these things. And so this is something that I learned from my PhD, that most of your communication has to take on an hourglass structure. A broad question, pointed, a broad field process, biological process, a pointed question, how do you answer it? What have you found? What are the implications? If you incorporate that in a lot of your communications, I think it's a very powerful way to do things. The other important thing, I was just telling somebody this, if you give two people two identical bits of gold, one wrapped in newspaper and one in very nice wrapping paper, chances are he will pick the one in wrapping paper.
True.
Though the gold is equivalent. Your research may be equally valuable and beautiful and valuable. Unless you are able to communicate it, to wrap it in words or a form that people are able to accept it, it will not realize its full potential and value.
Absolutely.
So for every audience, you need to learn how to sort of wrap things. And I actually look to podcasts, to written science articles, to movies, as to the structure in which things are communicated. Storytelling is a critical skill, and you have to learn from whatever is available to you.
Absolutely. I think I cannot at least put them in the way Dr. Dhananjay has just put. And so this is for all the audience that be trained in storytelling on the basis of who are your audiences, prepare your speech or, explanation, because the details differ on who is your audience at that point of time. So now we are at the edge of our conversation for this. Dr. Dhananjay, I would like to ask you, do you believe that researchers are celebrities?
Researchers, but by standard definitions, no. By the standard definition, no, because we have some fantastic scientists that do not get recognized outside their workplace. So I think, so by standard definitions, researchers are not celebrated in the same way as people who you see in movies are. But are researchers worth that much celebration? This is a very philosophical topic. Is anyone worth anything? I don't really know. But what I can say is that I don't know about the researcher specifically. I think knowledge and the way of thinking is critical. We have always had representatives amongst people of how to live, how to think about society, about nature, and one should pay attention to what they say.
Nice. And so here, the idea of celebrating research at researcher celebrities. So empowering science believe in breaking the barriers, and one of the barrier which you just figured out, like you just mentioned also, that researchers are not celebrated. So our token to the society, to the community of researchers, because speaking for myself, I don't think I can do anything except research. So we thought like, okay, let's start celebrating researchers globally, because they do need more than, they are the real life heroes, which should be celebrated more than, or, there is no comparison between real and real life heroes, but they should be celebrated. With the idea of this, today we are honored to have you as our researcher celebrity. We wish you all the best. Even if you call yourself that you are an infant as an PI, we see a mature PI, and we wish you all the very good luck for your future research.
Thank you very much. I'm very flattered to be on this researcher celebrities thing, because there's much smarter people everywhere. So, but I think the main idea, the thrust of your effort at ensuring that science and people who do science get some amount of one-on-one time with public is laudable. So thank you very much for having me. Appreciate it.
It was a pleasure having you, Dr. Dhananjay. And now, time for audience. If you want to write to Dr. Dhananjay, you have seen, as I promised in the initial starting of my conversation, that he says something, okay, but you have to use your head, your brain, that he's way more than having a great communication expert and a brilliant scientist. So if you need some guidance, or be it a philosophical also, because I had some interactions with him, so I can tell you that. So feel free to write it to him. If you want Empowering Science Foundation to put you in contact with our researcher celebrities, please feel free, write it to us. and never give up on research, because that's what we are good at. Thank you, and see you all in the next episode of Researcher Celebrity. Thank you, Dr. Dhananjay.
Thanks. Thank you very much, Raja.